Welcome. And it is with great pleasure that I introduce today's physiology seminar speaker, Mark Wu. Mark is actually originally from Houston, so I just learned that he's a native Texan, born in Austin. So, like probably he's more Texan than everybody else here. But anyhow, so after he was done with the high school, he did his undergraduate at Cornell and returned to Texas for MD PhD at Baylor. And as a graduate student, he worked in Hugo Bellin's lab, characterizing molecular mechanisms that regulate neurotransmitter release using Drosophila neuromuscular junction as a model. And after he finished the medical school, Mark did his residency at UCLA, then moved on to doing his postdoc at UPenn. And during his postdoctoral training, Mark was affiliated with Larry Zipursky's lab at UCLA and Amita Segal's lab at UPenn, and worked on dissecting molecular and genetic mechanisms that mediate sleep using Drosophila as a model. And Mark started his lab at Hopkins in 2009 and has been a professor of neurology, medicine, and neuroscience since 2019. So Mark has actually run a very prolific lab focusing on understanding molecular and cellular mechanisms that mediate innate behavior and homeostatic drives, including sleep and hunger, and using flies and now mice as model organisms. And Mark is also a sleep medicine physician, and so he has been studying the relationship between sleep and Alzheimer's, both in patients as well as in models such as flies, which I think is very much of a unique combination. And Mark has received numerous honors, including Burroughs Wellcome Career Award and Wayne Henning Investigator Award. And just a little side note, I actually overlapped with Mark when I was a junior graduate student in Zipursky's Zipursky lab. And one thing I remember vividly is how he presented his lab meetings. And Mark always presented his lab meeting as if it had been a job talk, with all the panels labeled, all the statistics done. And I remember I was very much impressed. And I learned that from Mark that every job talk is a job talk. Okay, so I'm very much excited to hear what Mark is up to. And so without further ado, please join me in welcoming Mark to UT Southwestern Physiology. Welcome, Mark.
Thanks, Daisuke. Thanks for that very nice introduction. Way to set the expectations very high for this talk, but no problem. So yeah, I remember overlapping with Daisuke back in, I guess this would be in the early 2000s. I was at that time a neurology resident. So during my elective time, I spent all of it in Larry's lab, which still wasn't that much. I was only there probably in total for like maybe 6 months or something like that. But it was always fun. It was a fun group. And I remember being very impressed by Daisuke back then. Even he's a graduate student. He was very bright, very hardworking, and very motivated. And I knew that he would go far. All right, so let me go ahead and share. Hopefully I can share my screen. Let me try here. Okay, are you guys able to see my screen?
Yes, we can see.
Okay, and let me start the slideshow. All right, so as Daisuke was saying, my lab studies the molecular and circuit mechanisms underlying innate behaviors, and we have a particular focus on sleep. And as Daisuke said, my lab traditionally used fruit flies as a model organism, but increasingly we're using the mouse as a model organism. And all of the work I'll tell you about today is unpublished and focused on a mouse gene that we've named wide awake and its role in regulating arousal. Okay. So as all of you know, most of the animals on our planet, their lives are shaped by the daily rhythms generated by the rotation of the Earth on its axis. In order to anticipate these daily fluctuations in light and temperature, animals have evolved an internal biological clock. And so through the seminal work of people like Joe, who's here at UT Southwestern, and other Drosophila circadian biologists, we've really unlocked the basic molecular mechanism of the core clock, as you all know, which is essentially a transcriptional translational feedback loop. In this example, which is for mouse proteins, the transcriptional activators clock and BMAL drive expression of a bunch of genes, but some of which include these transcriptional repressors, such as the period genes. The levels of these transcriptional repressors go up and then they go back to inhibit the function of these transactional activators, leading to their own decrease in expression, leading to these 24-hour rhythms. So obviously, the work over the past several decades has really led to a very detailed understanding of not only the nature of this core clock, but also how this clock is regulated, is stabilized, and entrained. But what is less well understood are what are the output mechanisms acting downstream of the clock to regulate various physiological behaviors. So as you all know, animals, including humans, have many physiological processes and behaviors that are under circadian control. You know, hormonal, things like hormonal secretion, things like arousal, things like body temperature, and of course, one of our favorite behaviors, sleep. So to study how the clock regulated sleep, back around 2009, we had conducted a large-scale genetic screen in Drosophila. And so I just want to introduce the fruit fly work related to this novel gene called wide awake, or at least back then novel, now it's not novel. I'm only going to summarize this work in model slides because all of this work is published. This work was led by a former postdoc in the lab, Sha Lu, who you can see here, who now has his own lab at the VIB in Belgium. So anyways, what we did a large-scale genetic screen and identified a novel fly mutant, a novel at that time, which we named wide awake, because these flies had very hard time falling asleep. So they had a prolonged sleep latency. And as a sleep medicine physician, it reminded me of my patients with sleep onset insomnia. And we named it wide awake, kind of like these insomniacs laying in bed, laying wide awake, unable to fall asleep. And so to summarize the model, basically what we found is that this gene called wide awake, or this protein called wide awake, acts downstream of the clock in arousal promoting clock neurons, and its levels rise at night. And what it does is upregulate the GABA A receptor, which in Drosophila is called RDL. And by doing so, sensitizes these arousal promoting clock neurons to inhibitory influences at dusk. Thus, when you lose wide awake, you reduce the expression of this RDL or GABA A receptors. You lose the GABA A evoked currents, and they cannot fall asleep as easily because these cells are not inhibited. And so that was kind of what we first showed back in 2014. And then, and then we followed that paper up by showing that wide awake does not only regulate the timing of sleep onset, but also it regulates sleep quality. Okay, and so this work was conducted by a former postdoc in the lab, Masashi Tabuchi, who now has his own lab at Southwestern. This model is a little bit complicated and slightly unrelated to what we're going to be talking about today, but I'd like to go through it because I think it's quite interesting. And so, so when you take a step back and think about neural codes, most of us think about rate codes. Rate codes are basically the idea that information is encoded by the frequency of neuronal spiking. However, there are other kind of codes. For example, temporal codes. Temporal codes are the idea that the pattern or timing of spiking matters. But they are much more difficult, in general, especially temporal pattern codes, have been difficult to study because they're often commingled with rate codes. And what Masashi found was that in these clock neurons, in the middle of the day versus the middle of the night, that the pattern of the clock neuron firing changed. That is, that during the middle of the day, the pattern of the firing was irregular. During the night, the pattern of the firing was regular, but the rate was essentially identical. And so he used this model to basically try to understand the function of temporal coding. And basically what Masashi showed was that wide awake, not only functions at dusk, but to regulate sleep onset, but also acts in the middle of the night to improve sleep quality by promoting more consolidated sleep rather than fragmented sleep. And so what he basically showed was just changing the pattern of firing alone could change behavior in Drosophila, specifically sleep quality, which is one of the first times that people have been able to really show specific changes in pattern of temporal codes or temporal pattern codes changing behavior. What he also showed, which is really neat, is that this wide awake gene, now and remember in the last slide I showed you that wide awake upregulates GABA A receptors. He showed that it also upregulates calcium activated potassium channels and sodium potassium ATPases. And the function of change in a dynamic way, again, upregulation at night. And by doing so, he showed that it actually changes the more biophysical properties of the spike. Most of us think of spikes as a spike is a spike is a spike. It's 0 and 1 coding, right? But actually, that's not entirely correct. Spikes are actually different. And what he showed is the actual nature of the spike changes between day and night. And these changes in the biophysical properties of the spike, faster slope upwards and deeper after hyperpolarization, lead to these regular pattern firing. And all of this is under wake control. Wake regulates these ion channels, wake regulates these spike properties, and wake regulates the pattern of the firing. And then he showed, really interestingly, that this pattern of firing is transformed into a rate code in a downstream arousal circuit. For the Drosophila people, these are PI neurons. And what's really cool is that it generates plasticity. The irregular pattern alone, in the absence of any rate changes, induces plasticity of this downstream arousal circuit, which is the first time that's ever been shown to our knowledge. So anyway, this is a little bit to the side of what I'm going to be talking about today, but I just wanted to go over because I think it's kind of a neat model. And so you might ask the question, okay, so we basically showed that wake acts in clock neurons under clock control to regulate various channels and receptors, to regulate sleep onset using a rate coding mechanism, and to regulate sleep quality using a temporal coding mechanism. And so you might be asking, why such a complicated mechanism, right? Why is, you have all these different coding mechanisms to regulate sleep. And of course, we don't know the answer for sure, but we have a hypothesis, and that has to do with energetics. The most expensive thing that a neuron does is spike. When you go from the wake to sleep transition, this is a brain-wide change that needs to occur relatively rapidly. And so that's why we believe, or we hypothesize, that the system uses rate coding, which is very strong, but very energy intensive. In contrast, sleep quality is a milder phenotype to manipulate and occurs over a very long time point. Instead of just at one time point, the wake and sleep transition, now we're talking about over hours, how do you maintain sleep quality? And it turns out that temporal coding is a very efficient means of coding. Why? Because the frequency of the spiking is exactly the same. So you don't have to use any energy for that. You just have to change the pattern of the spiking. So it's very energy efficient and allows, but yet is not maybe super powerful. And so that's why it might be well suited for tuning sleep quality. Okay, anyway, so that's kind of the backdrop now. I've now set up this is all published data. This is the backdrop for our mouse work. So the mouse work was spearheaded by a talented graduate student named Ben Bell. And basically what happened was there was one mouse homolog called ANKF1. It's pretty similar to the fly gene, although you can see that it lacks this unstructured domain. It has 2 anchor repeats followed by a fibronectin type 3 domain. And similarly in humans, there's only 1. And so as a fly person, that was appealing to me, right? Because as a fly person, we're always like, oh gosh, in mouse, there's always 4 copies of every gene. But in this case, there was only 1. So that was kind of enticing. The next thing that was enticing was that it was expressed in the suprachiasmatic nucleus, which, as you all know, is the circadian pacemaker system in mammals. And of course, we had been studying the role of wide awake and circadian timing in flies, and specifically in the clock network. Actually, a funny story. So Shah, the postdoc I mentioned before, who's now at VIB, was once giving a talk at Hopkins about his fly work. And one of the faculty asked him, Shah, where is the mammalian homolog expressed? And Shah said, the SCN. And my mouth just dropped because I was like, we did not know that at the time. And I said, after the talk, I said, Shah, why did you tell them that it was expressed in the SCN? Because that's exactly what they would expect, but we don't know that. And then Shah paused and he said, oh, I meant to say CNS. So anyways, one letter change can make a big difference. But it turns out that Shah was right. So at around this time, when I was looking at this is back in the early days of the Allen Brain Atlas, when I don't know if you guys had this experience, but I could not figure out how to use it. Like I was trying to look in the Allen Brain Atlas, and I just couldn't figure it out. And I kept trying. And then one day they kept changing the interface, right? And then one day I could suddenly use it. And then I looked at it, I was like, that looks like the SCN. Anyways, so then we decided to move into mouse because there was a single homolog in mouse and it was expressed in the SCN. So if you don't move into mouse at this point, then when would you ever move into mouse? So the next thing we did was we did a full brain-wide analysis of M-wake, what we call M-wake. And we used 3 different techniques. The first technique, and this is published last year in JCN. First, we used RNA-scope. And then second, we used transgenic mouse, which has a V5 insertion at the end of the gene. And third, we made another mouse, which has a TD tomato Cree insertion replacing exon 5. And let me just take a step back. Why did we do this? Because we failed to make an antibody. So I tried in flies, between me and a collaborator, we made like 12 or 15 different antigens to make a fly-wake antibody. We failed in all of them. In mouse, we tried to make, I think, 5 different antigens or 6, and we failed on every single one. Everything works for Western, but they all fail for immunostaining. Much later, I realized one of the reasons for the failure is that this gene has very low expression, actually. In fact, and I was telling Joe this earlier when I was meeting with him, you cannot detect the expression of this gene if you use 10x genomic single-cell sequencing. You can only detect it using SmartSeq, which is very annoying to us. Anyways, so this is how we assess the expression pattern using these 3 different tools, and basically they all show the same thing. I'll give you an overview of the expression pattern of the brain later, but for now, let's focus on the SCN. So using M-wake V5, TD tomato, and RNA-scope, M-wake is expressed in what's called the core region of the SCN. And I'll show you a little bit more. Actually, I'm not going to show you more details now, but the core region is the region of the SCN that in very broad strokes is thought to be photorecipient. If you're interested, I can tell you more about its expression later. Okay, so because in flies, right, we had studied the role of wide awake in the circadian regulation of sleep. So the first thing we did in mice was we made a knockout. So we made a CRISPR-generated knockout by engineering a stop codon in exon 4, which is seen in all isoforms of M-wake. And then we did EEG, right, in mice. And basically, we saw a very subtle phenotype. If you look at night, there's a little bit of reduction of non-REM sleep, but it wasn't super dramatic or anything. And so initially, we were a little bit disappointed that the phenotype seemed kind of not that strong. And then we remembered that mice, unlike flies, are nocturnal, and that wide awake tends to function at night, right? But mice are not sleeping, their main sleep period is the day. So we decided to actually look at arousal. So I don't know if this movie is working. It's kind of working, but it's very laggy. I don't know if you can tell, but one of the mice in this movie is an M-wake mutant, homozygous mutant. Okay, and if you can't tell, it's the one that's kind of running circles right now, basically. And here's another example of what these mice do. And so what you can appreciate is they're kind of spinning, right? I should mention that another team at UCSD, Bruce Hamilton's group, also independently identified ANKF1 and they were thinking that this was like a vestibular mutant because they were looking at sort of things that did the spinning kind of thing. So I can go into the details if you'd like later, but we do not think that this phenotype is vestibular in nature for a variety of reasons. But rather, we think it's a stereotypy because you can actually induce this phenotype by, for example, giving mice stimulants or by making their brains hyperexcitable. But for those that are interested, we can discuss that a little more in detail later. So basically, what we found was that unlike our sleep phenotypes, the locomotion phenotypes were very significant. And so this is a quantification of that. We put these mice in these beam break cages where basically you have these infrared beams and these, and a computer counts the beam breaks, right? And this is a control mouse under DD conditions, which means constant darkness. And as you would expect, mice move around more at night because they're awake at night, right? And this is what happens in the mutant. The gray is the same data I just showed you, but it just got scrunched because of the scale totally changed. And these are the mutants. And what you can appreciate is the M-wake knockouts are really active at night, okay? And they are more active during the day, but really it's not as impressive, right? Because you can see this curve compared to this and this compared to this is really different. I should say that this phenotype is, you can see the error bars are pretty big here. And that's because a substantial part of the phenotype is driven by about 30 to 40% of the mice, which spend a lot of time circling. If you remove the circlers, they're still hyperactive, but not as hyperactive. Okay, so we started to realize that maybe in mice, wide awake is not regulating sleep so much, but regulating arousal. So what are some other measures of arousal? Besides locomotion, you can also look at sensory responsiveness, right? And so we did this by doing acoustic startle within the mice. You just give them a tone and you just look and see their jumpy response, right? We did this for controls versus mutants. I should also say for a lot of our assays, I'm not showing the data, but we've also done like heteroallelic combinations and second allele, so it's not just one allele that we're looking at. But I'm just showing one allele for simplicity. Anyways, so what you can see is during the day, there's not really that much of a difference between the controls and the mutants. But at night, and this is subjective night, you can see there's a pretty big difference in the sensory responsiveness at 100 decibels, and also a significant response at 110 decibels. And then as it keeps increasing the volume, the difference kind of goes away. It's almost like the mutants kind of saturated from the beginning. Okay, so I didn't show you this data, but we previously showed in Drosophila that the clock neuron firing pattern changes in wake mutants. That is, that you guys remember that in clock neurons, they typically fire more at the beginning of the day, and they fire less at the beginning of the night, okay? And in flies, what happened was that when you lost wake, that rhythm change was lost such that the fly clock neurons fired more at the early night. And so basically the same thing happens in mouse. So we used our TD2, so this mouse that we made that basically replaces exon 5 or 4, I can't remember which exon this is replacing, but it puts in a TD tomato 2A Cree, okay? So it basically generates a mutant, it labels the cells, and also provides Cree access. And so what happens is this work was done by Chong Lu, a postdoc in the lab, and he did slice recordings of the SCN and recorded from M-wake heterozygous, which we're considering controls, and M-wake homozygous Cree, which are basically mutants. And what you can see, as expected, in contrast, in the mutants, they have high firing both day and night, which you can appreciate. I think I froze for a little bit, so I'm gonna repeat my last two sentences. So in the mutant, you can see that there's high firing during the day and the night. So you basically lose the rhythm of firing, okay? Again, the way to think about how wake works both in flies and mice is that it tends to upregulate inhibitory channels and receptors at night to suppress excitability. That's generally what we see, okay? And these data are supportive of that concept. In addition, remember in Drosophila, we showed that wake upregulates GABA A receptors and reduces, therefore, GABA A evoked current. And we see the same thing in mice. That is that if you take M-wake SCN neurons that are control versus mutant, in the mutants, the GABA evoked current is significantly reduced. So basically, we think that the basic function of M-wake is conserved between flies and mammals. That in both cases, wake is acting mainly at night to upregulate GABA A receptors and potentially other receptors and channels to reduce the firing and excitability of those neurons at night. But unlike flies, which are kind of diurnal, I mean, technically they're crepuscular, but unlike flies which sleep at night, mice are awake at night, And so instead of regulating sleep, what wake does is regulate arousal at night, basically. Okay. So remember that I mentioned that we did a brain-wide expression analysis of M-wake. And the reason why I bring this slide up now is because unlike the fly situation, where we showed that a lot of the arousal, the circadian related sleep phenotypes could be traced to the clock neurons, we found, and I'm not going to show the data because they're negative, that in mouse, M-wake does not seem to act in the SCN to regulate arousal. So we had to move outside the SCN to figure out where this was happening. And so first, I just want to, I realized that maybe on Zoom you can read this, but normally in a lecture you would not be able to read these little letters, but I'm just going to very briefly summarize this. M-wake is expressed in a distributed yet restricted manner. So it's not everywhere, but it is kind of throughout the brain in multiple specific locations. The most dense location of M-wake expression is in the hypothalamus, as we discussed in the suprachiasmatic nucleus, but also in the dorsal medial hypothalamus, the ventral medial hypothalamus, the zona incerta, the TM, near the TMN, the tubermammillary nucleus, the lateral hypothalamus. It's also expressed in some brainstem nuclei, like the locus ceruleus, the parabrachial, which have been implicated in arousal. It's also interestingly expressed in various components in the limbic system, like the lateral amygdala, the central amygdala, the basomedial amygdala, the bed nucleus striata terminalis. It's also expressed in a number of sensory regions, like the olfactory bulb and some auditory and vestibular nuclei, which is why Bruce Hamilton was really interested in this vestibular idea. So among all these regions, the dorsal medial hypothalamus in particular has been implicated in circadian regulation of arousal. So we decided to next focus our attention there. So specifically, Jun Liu's group at Harvard and Cliff Saper's group at Harvard back in 2003 showed that if you ablate the dorsal medial hypothalamus, so you just take out the whole thing, that you can actually suppress circadian regulation of arousal or clock-driven arousal. And that here I'm showing a plot that looks at locomotor activity. And so you basically see that overall, instead of having a rhythm, it's kind of overall activity is reduced and it's kind of flat, see? In addition, Gary Aston Jones, around the same time, published in Nature Neuroscience a circuit that goes from the SCN to the DMH, dorsal medial hypothalamus, to the locus ceruleus, which, as you know, is noradrenergic. And what he found was that the firing of these locus ceruleus cells is under sort of like clock control, such that there's more firing during their wake period in the night, and there's less firing during the day period, their inactive period. And that if you lesion the DMH, that you lose that rhythm of firing in the locus ceruleus. And so basically this is what led us to move into the dorsal medial hypothalamus next. So the first thing we did is we decided to knock out M-wake specifically in the dorsal medial hypothalamus in an adult specific way. To do this, we generated a floxed allele of M-wake, where we basically put LOX-P sites around exon 4, and then we injected AAV-Cre into the dorsal medial hypothalamus, which is shown here. And basically what you can see is that when you do this, you get a significant increase in locomotor activity at night. You get a little bit of a trend towards this during the day, but it's not significant and it's much more variable. And of course, sham doesn't really do anything. So this led us to start focusing on the DMH circuit with the M-wake positive DMH circuit. The next thing we did is do patch clamp recordings of M-wake DMH neurons from sliced tissue, as we described before for the suprachiasmatic nucleus. So these are M-wake positive DMH neurons. These neurons also show rhythmicity in their firing. Interestingly though, instead of firing more during the day and less at night, they actually fire more at night, which follows the mouse's nocturnal pattern. Okay, and what happens in the M-wake mutants? Remember that the job of wake in general is to inhibit the excitability and the firing of neurons at night. They actually are even more increased, basically. And so you can see that in M-wake DMH neurons, during the day, their firing frequency, this is spontaneous mean firing rate, doesn't change too much, but at night, it substantially increases, okay? So the next question is this a cell autonomous function? Because in that case, you could have other things going on. And so we looked at intrinsic excitability. And it turns out that yes, it appears, as we expected, it is a cell autonomous function. I mean, this is also, to be honest, we knew this because this was also the case in fly. We had already known this was the case in fly. But basically what you do, these are FI curves where you inject current and look at the frequency of the firing in the cell, and you clamp the cell. And what you see is that during the day, in the morning, there's really no difference in intrinsic excitability between controls and the M-wake mutants. But at night, you can see that there's an increase in the excitability of these M-wake DMH neurons. Okay. So next, we wanted to look, so this again, all of these data are basic. So far, this is saying that, okay, well, if we knock out M-wake in the DMH, we see more activity specifically at night. And when we knock out M-wake, we see increased excitability and increased firing of these cells. So it all makes sense, right? However, this was done in sliced tissue. So we wanted to next look in vivo. So to do this in vivo, we did fiber photometry next. And so basically what we did is we took a mouse that's M-wake Cree, and then we implanted a fiber, we did fiber photometry. Well, first we injected, AAV flex GCamp, right, into M-wake Cree. And then we planted the fiber into the DMH. And then we also put EEG on there so we could know what behavioral state they're in, okay? And if you focus here on the bottom left, basically these are the mean delta F over F for the calcium signal. And QW stands for quiet wake, NR is non-REM, R is REM, and AW is active wakefulness. And what you can see is this is day and this is night. And I've made the scales the same so you can directly compare them. What you can see is that there's a big increase in calcium activity in the M-wake DMH neurons during both active wakefulness and REM sleep. And it goes down at quiet wakefulness states and non-REM. And during the day, although there's a bit of a trend that direction, the amplitude is quite small. And in fact, none of this is significant. So this is consistent with our data, with our slice, ex vivo ePhys data, right? Because activity is high during these, but it also provided some new information, which is that we did not know that these cells were active during REM sleep. We next did transition analysis, where we basically looked at all of these active wake to non-REM transitions, non-REM to active wake transitions, and non-REM to REM transitions. And we did this during the day and the night, okay, dawn and dusk. And basically what you can appreciate is that the transitions, the activity of these cells change as you would expect. When they go from active wake to non-REM, it goes down. When it goes from non-REM to active wake, it goes up, right? And non-REM to REM. And that we see similar trends in the morning, but that at night, these, again, the amplitude and also the slopes are sharper. The amplitudes are bigger and the slopes are sharper, but it's basically the same deal. So basically these fiber photometry data suggest that the activity of these M-wake DMH neurons are increased during REM sleep and active wakefulness. We next looked at transition, number of transients. So transients are basically like, and here's an example, if you look at a fiber photometry trace, you basically get these little peaks, right? And these triangles are pointing to what our algorithm was calling as a transient. When you do this analysis, what you see is that there's a significant increase in transients, specifically in active wakefulness. And at night, it's pretty clean in that everything else is pretty flat and the active wakefulness transient number is higher. In the morning, there is also an increase in number of transients in active wakefulness, and strangely, curiously, a decrease in REM. I don't know, we don't really know what that means yet. Okay, so now I've told you a little bit about the activity of the M-wake DMH neuron. So next we want to look at the function. And so we next move to using DREAD to functionally manipulate these neurons. So the first thing we did was basically take the M-wake Cree mice and inject floxed, or DIO, DREAD GQ to activate these neurons. And we injected the virus into the DMH, of course. And this is just to show you we infected the DMH. And basically what happens is then when you feed them CNO, you get a lot more activity. Okay, so they're hyperactive. Okay. And then if you look at EEG, what happens is basically you induce, when you activate the M-wake DMH neurons, you induce a profound wakefulness. This is control. So these are the same mice that are M-wake Cree injected with virus that is a FLOX-DREG-GQ into the DMH. And if you inject vehicle, basically this is their natural rhythm. They're kind of sleepy. You know, they're not that awake during the day period and they wake up at night, right? But when you inject CNO to activate the M-wake DMH cells, they're basically awake the entire time. They don't sleep. So these data suggest that if you activate the M-wake DMH neurons, that it produces increased arousal, right? Because they have increased wakefulness and increased locomotor activity. So where could this be acting? So to start to address this, Sang-Soo, a postdoc in the lab, just tried to inject AAV flexed GFP into M-wake Cree mice, and he injected into the DMH. And what you see is basically this. So he injected the DMH, and you get projections that go all over the brain. And here's a zoom up that's a little easier to appreciate. So again, these are all starting from M-wake DMH positive neurons. And you can see fibers that run all over the brain. You can see fibers that run, you know, through the caudate here, that go this way, that even go all the way almost to this white matter tract up into the cortex. You can see a lot of fibers that go back and actually seem to touch the locus coeruleus, which we talked about earlier, which is a known arousal center, and also go down the brainstem surrounding the pons and various areas down here. So because we were aware that the locus coeruleus is an arousal promoting area and that Gary Aston Jones had suggested that the SCN can, you know, via the DMH communicate with the LC, we decided to look to see whether these M-wake DMH positive neurons also could connect with the LC and manipulate them. And so basically what we did was we injected channel rhodopsin, flex channel rhodopsin into the M-wake Cree mice, into the dorsal medial hypothalamus. And then we shined blue light in the area of the LC just to activate those terminals, right, that project to the locus coeruleus. And by doing so, we can show that activation of these M-wake DMH neurons leads to firing in the noradrenergic locus coeruleus cells. I should say that we do not know at this point whether this is a direct or an indirect connection. We have not done those experiments yet, using, for example, synaptic blockers, et cetera, to kind of prove that, or tetrodotoxin, I mean, to show that it's a monosynaptic connection. But what we can say is that there's a functional connection between M-wake DMH neurons and noradrenergic locus coeruleus neurons. So next, let's silence these neurons. So the way we did this was we used M-wake Cree mice, and we injected FLOX-dependent DREG-GI into the dorsomedial hypothalamus. And then we fed them CNO. And basically what we see, and this is on here in the middle, is a spectrogram, which shows you basically a heat map, the intensity of various frequencies. On the y-axis is frequency going from 0 to 40 hertz. On the x-axis is time. Here you have the day, and here you have the night. And then this is the heat map here, okay? And here what you can see is basically the REM state. And basically what happens is that in control mice, where you just inject saline instead vehicle, right, instead of CNO, okay, into the same mice, this is the REM sleep that they have. But if you inject CNO to basically silence the M-wake DMH neurons, you basically had a marked reduction in REM sleep. And remember that these neurons were active during REM, right? Okay. But we were wondering whether M-wake DMH neurons, we think that they also are involved in arousal, but we didn't see an arousal phenotype here, right, in the silencing experiment. However, as many of you know, the arousal system in mammals is quite redundant. And this has been shown over and over again, that if you take out a single arousal circuit in mice, that you often have no effect on sleep-wake. And that often it takes the knockout of multiple circuits to have this effect. So, and these data are preliminary. We literally generated them like a week ago. And so I only have n equals 3, but I just wanted to show you because it kind of is related to the story. So what Sang-Soo did is take M-weight Cree mice and injected and did kind of a multi-circuit injection or multi-site injection, we call it, of flexed DREAD GI. Again, this is a chemogenetic way of inhibiting these neurons, and did this in 2 locations, in the hypothalamus, okay, but also in the kind of dorsal lateral pons area, which contains more arousal circuits. And basically, if you do this, what happens is that you do get a reduction in locomotor activity. You do get a reduction in, apparent reduction, let me say, in wake state. But again, I'm not showing statistics here because this literally is an n equals 1 experiment with 3 mice. So, we're now repeating this now, but we think this will probably pan. In fact, I think Sang-Soo told me this, he already reproduced these data, but I just don't have the updated plots yet. So the idea being that if you silence the M-wake DMH neurons, but a little bit more, then you can suppress wakefulness. Okay. So we also did single cell sequencing of M-wake cells in the mouse brain. And the way we did this was, like I said, this is a little bit challenging, right? Because M-wake levels are so low that it's hard to detect even by single cell sequencing. So the way we did this was we took our M-wake TD tomato Cree mice that expressed TD tomato in M-wake cells, and then we dissected the hypothalamus out. We FACS sorted the TD tomato positive cells, and then we sequenced the TD tomato positive cells. And then basically, we use informatic approaches to then map them to certain anatomical locations. And what you can see here, and this is basically these, plots, UMAP plots, and then this is like a heat map that gives you expression of various genes in various clusters. And the bottom line is that M-wake in the hypothalamus, and we also can match this with our expression analysis as well, right? So M-wake is expressed in a number of clusters in the suprachiasmatic nucleus. I was talking to Joe about this earlier. And for those of you that like thinking about the SCN, M-wake is expressed in GRP positive neurons, in VIP positive neurons, but not in AVP neurons. It's also expressed in some other clusters like ROR-B positive neurons that are neither GRP or VIP positive. Anyways, but M-wake is also expressed, as we just have been talking about, in the dorsal medial hypothalamus, near the TMN. There's some cluster of the VMH and the preoptic area as well. So what I want to draw your attention to is there's 2 clusters in the dorsal medial hypothalamus, which we're just calling cluster 4 and cluster 0. It doesn't really matter. One of these clusters is galanin positive, and the other is galanin essentially negative. But again, it's kind of a continuum of expression, but it's basically gal positive, gal negative. So the reason I bring this up, and then the gal positive cluster 4 is largely glutamatergic, although there are some GABAergic cells. The reason I bring this up is that Yang Dan's group has found that there are some DMH, dorsal medial hypothalamus positive cells that promote REM sleep that seem to be galanin positive, but her cells are GABAergic. So we're not exactly sure yet what is going on, but we hypothesize that there are obviously more than one subgroup of M-wake DMH cells, and that the regulation of the REM sleep and the arousal will be mapped to different subsets of cells. We are doing these experiments now by using double intersectional approaches, using FLIP-FRIT with Cree-locks. Of course, this is much more annoying in mice than in flies because each cross takes so long. So this is we're in the crossing, and it'll take us probably till summer or late summer before we have this data. But what we hope to find is that one of the clusters may be involved in arousal and one may be involved in promoting REM sleep. Okay, so let me just summarize with a model kind of what we've been talking about today. So in mouse, what we've been talking about is the circadian regulation of arousal. And we all think about how the suprachiasmatic nucleus is important for all kinds of rhythms, including arousal, right? So if you ablate the SCN, you lose rhythms of arousal, right? And there is data from other groups suggesting that the dorsal medial hypothalamus may be a relay between the SCN and regulating arousal, or may be important for this process. What I've talked about today is a clock output molecule that we're naming M-wake that we think functions in a cell autonomous way within specific M-wake positive DMH neurons to provide local regulation. Because we all know that there are not only this sort of master pacemaking mechanism, but that as, because Joe is at Southwestern, you all are very intimately aware that every cell has a clock, right? Every cell has clocks, and these local clocks may also serve an important function. And so we think that M-wake is a mechanism by which local DMH clocks also amplify the rhythms of arousal. And interestingly, also REM sleep. And so we're trying to tease apart the exact circuit mechanism of this, these two pathways now. But that's kind of the basic, overall take-home message. To summarize what I've told you today, wide awake is a clock output molecule that regulates sleep in fruit flies and arousal in mice, because mice are nocturnal. So we assume that in humans, wake would also regulate sleep, actually. And in that way, mice, as you guys know, is sometimes a funny model for sleep, right? Because they don't make melatonin, and they're nocturnal. But anyways, and in general, what happens is that in mouse, M-wake suppresses arousal at night, likely by inhibiting the excitability of arousal-related circuits at that time. And that's in general what we think wake does, is inhibit excitability of neurons at night. M-wake positive neurons in the DMH promote arousal and REM sleep, and there may be two distinct populations that serve these different functions. So I just want to thank the people who did the work. So I talked about Shah, who spearheaded the fly wake work, and he's again now PI at VIB in Belgium. Chi-Li Liu did a lot of work on the fly wake story as well, and she's now a PI at UCSF. Masashi Tabuchi, as I mentioned, who's at Case now, did the temporal coding story in fruit flies. Ben and Chang are the main drivers of the mouse wake story. Chang has done all the physiological experiments, the ePhys, the calcium imaging. And then we also have collaborated extensively with Seth Blackshaw's lab, who's helped us with the single cell sequencing. And then we also collaborated with Kachan, a computational neuroscientist that helped with this coding story with Masashi. Okay, so I think I'll stop there, and I'll be happy to take any questions.
Okay, great. Thank you, Mark. So we have time for discussion. If you have questions, either raise hand or put that in chat or Q&A, and I will unmute you. Okay, somebody.
Actually, could I ask a question? Okay, so basically what I'm wondering is, are there mutations in humans, or perhaps even polymorphisms in humans, that that could be found in all the sequencing of humans that have been done? And would there be, is there any way of searching to see if there's any correlation with, people who only require 3 hours of sleep or anything along those lines? Is there any way of doing that?
Right. So what I can tell you is that in humans, there are polymorphisms in this gene. They are not common, suggesting that they are selected against. So for example, if you go to Nomad, you can find, presumably deleterious coding mutations in this gene that are rare. We do not know, because it's Nomad, you don't know the phenotypes associated with those humans, right? Because those typically are control populations or general populations. One possibility, and I've been exploring this idea to try to get at this question, is the UK Biobank, because the UK Biobank, as you guys know, is about 100,000 people, right, that have had GWAS done. About 30 or 40,000 of them have had actigraphy done. So at least you can get some measure of activity. They don't have, obviously, EEG, because that can't be done on that scale, right? And what they are trying to do in UK Biobank is do exome sequencing on all 100,000. I think they're currently at like 20,000 or 30,000. So I think that would be a resource. And I applied for access to this database. And I think it'll be interesting once they get full exome sequencing to then look at those people that might have variants in ANKF1 and then look at their actigraphy, basically. They also have sleep questionnaires as well. But questionnaires, as you know, are subjective and always a little tricky to deal with.
Okay, I think Yang Yu, yeah.
Hi, thank you.
Hi.
I really enjoy your talk, and I have to say that I'm also suffering this 1 hour change that
Yeah.
happening right now. So I just want to make sure that whether I understood correctly. So I have two questions. So did I understand correctly that when you knock down this M-wake in mice, they are hyperactive, but when you also when you do the QDM, then you saw the hyperactivity too? Is
I'm sorry. I think you froze. So can you repeat the question? I didn't hear it.
Hello.
Hello. Yeah.
Okay.
Can you hear me?
Yes.
Can you hear me?
Yes. Yes.
Okay. Yeah. I froze. You froze, so I couldn't hear your question. Could you repeat the question?
Yes. So your dread Q experiment of DMH.
Yes.
And it also leads a hyperactivity of the mice.
Exactly. So that's right. So how do we explain that relative to the mutant? So it's actually exactly what you would expect. Because remember that the function of wide awake is to inhibit the cells that it's expressed in. So when you knock it out, those cells become firing more like you were using dread GQ.
Okay, so that's why you want to knock down the GI so then you can see the arousal phenotype. Okay, that makes a lot of sense. The other question is this a high activity links to any food intake or body weight phenotype?
Yeah, that's a great phenotype question. So we haven't done this in great detail. I was talking to Joe about his work in doing, feeding and aging and stuff. What I can tell you is that these mice eat a lot more. So we have not quantified this, but we just know from replacing the food that the mutants eat a lot more and they don't get fat.
Oh.
They're skinny. If anything, there's a little skinnier, I think.
Okay.
So I think it's because they run around a lot. They get a lot of exercise. Yeah.
Okay. Thank you so much.
Yeah, thanks.
I think, Mariko, are you still here? I can't find you.
Yes, but just a simple question. Are there any specific clock genes that are affected by this M-wake mutant mice?
Yeah, so we don't expect clock genes to be affected because we think it's downstream of the clock. So we think that it's an output from the clock that we presume will not affect clock genes. Although I should say we have not looked at that specifically, but that's our hypothesis or our model, working model.
Okay, I have a question. So in flies, the wake mutant fly, or I guess at nighttime, you observe more regular firing, right? That supports sleep quality.
Right, in the middle of the night.
In the middle of the night. And then that is also regulated by wake through the KCA and the transporter. Is that true in DMH as well? And if so, is the regularity of firing connected to activity, hyperactivity phenotype?
Right, so that's a great question, and we get that question a lot. So the short answer is we don't know yet. So the longer answer is that in the SCN, what we know is that the regularity patterns between day and night seem to be the same as flies. What does that mean? So the first thing to say is we recorded in the mouse SCN, we recorded from dawn and dusk, right? And if you look, in dawn, the firing pattern is irregular, and at dusk, the firing pattern is regular, because there's ways to quantify this, right? And that if you look in the M-wake mutant, it's irregular during the dawn and irregular at dusk, just like in flies. However, and we gave this data to some reviewers because they were, we're talking about this and whatever, but that experiment is imperfect because we did it at dawn and dusk. There's also the overlay of firing rate changes. See what I mean? Because the firing rate's also higher during the day. So it's not a clean experiment. The cleaner experiment would be to look in the middle of the day and the middle of the night in the SCN or the DMH. And we have not done that yet. But one of my students is interested in pursuing this, although it is it's challenging and a lot of work because you have to do a lot of ephys basically.
Great. Peter?
Yeah, hey, Mark. So it's great.
Hey, Peter.
It's awesome. Awesome work. Question for you. I'm curious about the stereotypy phenotype, whether you think that's secondary to the arousal, or do you think that M-wake actually has specific roles in circuits that might be impacting these stereotypies?
Yeah, so for those who don't know, Peter also and I grew up both together in Houston, and so kind of like family friends, and his parents were faculty at Baylor and were mentors to me when I was an MD Ph.D. student. So we have a lot of connections. And also he was like really best friends with my sister back in the early 80s, I think. But anyway, so yeah, so the short answer is we don't know. And it could be either. These mice are super hyper aroused. And so it could be that, because like I said, you can get similar sort of like circling if you just inject amphetamines into mice, right? But M-wake is, as I also showed, M-wake is expressed in a number of regions throughout the brain, including like in, limbic system and all kinds of places. So it's possible that that stereotypy could be circuit specific and not just simply a measure of super hyperarousal. We don't know the answer to that question.
Great. Maybe one last question from attendee. If not, I will get an honor of the host, and I'll ask one last question. So for the sensory arousal phenotype, do you think that is just all downstream of locus coeruleus, or is it actually like mediated by a different, like for example, olfactory bulb you said expresses and like, are there excitability regulation at many different centers?
Yeah, that's a great question. And so again, the short answer is we don't know. Our hypothesis would be that actually it probably is at multiple levels of the system. So, and the reason why I think that is that M-wake, the wake in both flies and mice seem to be expressed at multiple levels of a circuit. And we think one of the reasons that may be is to kind of strengthen or amplify rhythms of excitability along a path. In Drosophila, this is the case for different levels of the clock system. And so M-wake acts at different levels of the clock network we think to amplify the rhythmicity is that kind of concept. So we do not have any direct evidence for this at all. But our hypothesis would be that it is acting at multiple levels of the sensory system, like, more upstream and then more downstream to then kind of amplify response. These mice are really quite crazy, actually. They're very, you just touch them, they're different because they're super hyperreactive. They're very like, abnormal. And so it's important when we do our analyses to do them in a circuit specific way. And so that's what we try to do now. Yeah.
Oh, great. All right.
There's one more question in Q&A.
Oh, Q&A. Sorry, Teresa. Okay, let's see. Teresa says, for the reduction in REM sleep, was this due to a reduction in the number of bouts or bout duration?
Yeah, that is a good question. And I believe, and so I'm not 100% sure of the answer. I believe it's the number of bouts. Yeah, I believe it's number of bouts and duration, actually, because it was quite a marked decrease in REM sleep. It was almost nothing. But I have to say, I don't remember exactly.
Yeah.
Teresa, do you want to follow up on that question?
Yeah, I mean, I guess I was just wondering whether there was a lack of transitioning into REM sleep, or they transitioned normally but didn't stay in REM sleep. But it sounds like it was so reduced that you can't really tell because it was
Yeah, and also that might get into technical issues of scoring too, right? Because if you flip in and out of REM, it may not be easy to say that you're in REM.
Yeah. So what is your bout length that you're scoring?
I think that they're using standard, is it 6 seconds? And I don't score them myself. In humans, we score 30 seconds. And in mouse, I think they're using 5 or 6, whatever the standard is.
Yeah, so there's not actually a standard. I think.
No, I know. Some people do like 10, but I think that, but the one that's lower, like what is it like 5 or 6?
4 is kind of like
4, yeah.
It's
then
lowest that's commonly used, I think.
Yeah, it's in that, I can't remember if it's 4, but it's in that ballpark. It's less
Okay.
than 10 for sure, like 5 or 6 or 4, something like that. The only reason I can know that is because I score human EEG, and we do 30-second windows, and it's much less than that.
And then do you have like a criteria for like the number of successive epochs in order to begin a state, or?
Yeah, but again, I don't know myself the technical details of that because I don't score it. I don't remember what they're using, but we basically have tried to use what most people do in literature. But I understand what you're saying, that in the mouse field, it's not as regimented as the human field, where we have very strict criteria. And in the mouse field, people are a little more floppy, I think, about the definitions of things. But yeah, there are criteria, but we use some criteria, but I can't remember exactly what it was.
But you have the data so you could check to see if they're just very short or.
Oh, absolutely. Absolutely. For sure. For sure. It's there. We can go back. Again, it might be a little hard to score it if it's like super brief, but it's there. Yeah.
It's very interesting. Thank you.
Yeah. Thank you.
Okay, great. I'm not missing anybody else. If so, thank you so much, Mark, for the fascinating talk. So good to see you, and hopefully we'll see you again soon in person.
Yeah, it would be nice to see you all in person. All right.
Thank you.
Take care, everyone. Bye-bye.
Thank you guys.